Well, good morning, everyone, and thank you for attending the Quarter 2 scientific webinar sponsored by the American Statistical Association's Safety Working Group. We're very pleased today to be able to have Dr. Paul Hayashi, who will be presenting on emerging tools to support DILI assessment in clinical trials with abnormal baseline serum liver tests or pre-existing liver diseases. My name is Jim Buchanan. I'm president of Covalence. I'd like to introduce the rest of the speakers for today's session. Dr. Hayashi is the Associate Director of DILI within the Division of Hepatology and Nutrition at FDA. His research and publications have focused on drug-induced liver injury for the last 20 years. He was a co-investigator for the NIH Drug-Induced Liver Injury Network and remains the co-chair of the Causality Committee. Our panel today will be comprised of Dr. Barbara Hendrickson, who's on the faculty at the University of Chicago, and a former vice president of pharmacovigilance and patient safety at AbbVie. She also co-leads the Interdisciplinary Safety Evaluation Workstream and the Aggregate Safety Assessment Planning Task Force of the American Statistical Association Biopharmaceutical Safety Monitoring Working Group. And she's joined by Cindy Akshay. She is the Senior Director of Biostatistics at UCB Biosciences, where she leads the safety statistics team within the Biometrics and Quantitative Sciences group. She is also a member of the American Statistical Association Aggregate Safety Assessment Planning Task Force. And with that, I'd like to turn the conference over to Dr. Hayashi.
Well, thank you, Jim, for that introduction, and thank you for the opportunity to present some work we've been doing with abnormal baseline liver tests, but it's I put it broadly under this title, Regulatory Challenges in DILI Risk Assessment. This is our disclaimer. The content in this presentation is the responsibility of me, the speaker, and it does not necessarily represent an official view from the FDA. So this is my outline. I'll be covering EDISH a bit, talking a little bit about the origins and strengths, because to provide a basis of where we have to go from there. And we'll be talking about some of the failings and pitfalls related to EDISH, and in particular, the third sub-bullet there, trials with abnormal baseline tests. As Jim mentioned, I'll be spending some time on some new tools in that space. I will also cover some sort of broader deficiencies in trial protocols that are just we see fairly often, and I just wanted to bring them to your attention in terms of mitigating DILI risk and study protocols. So to start any really talk about DILI and regulatory work, we have to step back and mention Dr. Hyman Zimmerman. He was an internist and hepatologist and widely considered the patriarch of drug hepatotoxicity. He got his medical degree at Stanford and actually was all set to go into endocrinology. But like for many people around the world, the World War II interrupted his plans. He was drafted by the U.S. Army and was put in a field hospital around France. And there he took care of a lot of soldiers who, during an outbreak of hepatitis A, if you remember, this happened during the Battle of the Bulge. For those of you who may remember that was Germany's one of their last-ditch efforts to turn the tide later in the war, and they made the somewhat surprise attack on around Christmas to catch the Allies. Sleep at the wheel, and it actually did work for a bit. But during that time, there's a lot of snow around, and the GIs were using that melted snow to make coffee and get water and that kind of thing. And the water or snow was contaminated with hepatitis A. So with that, he just changed his career plans. He got interested in the liver and went on to have a very storied career around the DC area and at GW and the Armed Forces Institute of Pathology, as well as the VA. So it's sort of interesting. I always like to muse about what would have happened if things had been different and he'd been stationed somewhere else. I'm not sure where we would be. with drug-induced liver injury and how we handle it in regulatory work. So among the many things that Zimmerman is known for, probably one of the most famous, if not the most famous, is what is called Hy's Law. It's coined by others. Zimmerman that never really coined that term. In fact, I've heard through the grapevine he didn't really like that term. But anyway, he did write in his textbook, drug-induced hepatocellular jaundice is a serious entity. Mortality rate ranges from 10 to 50%, and this was just based on his astute clinical observation. Now, jaundice is fairly well accepted to be 2 times upper limit of normal bilirubin, but hepatocellular is a little less clear. But in his textbook, he mentions these enzyme parameters, hepatocellular being 3 to 50 times upper limit of normal, with an alk phos no higher than 1 to 3 times upper limit of normal. Now, around this time in the 90s, there were several drugs having trouble on the market. They were being removed and they were actually doing some significant harm to patients and some fatalities. So Congress got involved and the FDA was tasked to figure out how they wanted to handle this. And so a lot of this was based, what they did was based on Hy's Law. And the reason they reached for Hy's Law as a way to look into this problem is that the track record for Hy's Law has been really quite good. In DILI registries, at least 3 of them, it does come in right around 10% mortality or fatality rate across these 3. So as I said, when the FDA started looking back at the cases that had been removed from the market for troubles with fatal hepatotoxicity, they decided to look at Hy's Law cases, look for them in the applications. And sure enough, it was just a couple of Hy's Law cases that did appear in these drug applications for drugs that had troubles post-market. And I'll give you just 3 examples here. So you see, it doesn't take but 1 or 2 cases or a couple cases in fairly large registry trials to start predicting some problems in the post-market space. So from that was developed this statement that is now in the FDA 2009 guidance. Basically, the guidance suggests there's a low tolerance for Hy's Law cases, suggesting that finding 1 Hy's Law case in the clinical trial database is worrisome. Finding 2 is considered highly predictive that the drug has the potential to cause severe DILI when given to a larger population. And if you do this statistical work, there's something called the rule of 3, which means that you need a trial of about 3,000 exposed, not just enrolled, but exposed, to have a 95% probability of observing just 1 Hy's Law case. So again, a fairly low bar for at least raising some concerns about whether the drug's going to have trouble post-market. It became a task of these 3 and others, like Mark Avigan and others, to say, well, how are we going to find these Hy's Law cases efficiently if we don't want to miss even 1 or 2? And this is how EDISH was born. EDISH is just a straightforward scatter plot. You plot the peak bilirubin in times upper limit of normal by the peak ALT or AST along the x-axis. And every patient is plotted by their study arm. And you get a scatter plot. And then sort of the simple brilliance of this is they just decided to take this continuous variable output and overlay a categorical variable, which is these cutoffs, 2 times upper limit of normal for bilirubin, meaning jaundice. So everyone above this line technically is jaundiced or had a bout of jaundice. And then they set the vertical line at the ALT level of 3 times. This is the lower limit of ALT in Hy's Law textbook on hepatotoxicity. He said 3 times to 50 times. So putting these 4 familiar quadrants together, this becomes the familiar potential Hy's Law quadrant. Anybody up here draws our attention to potentially meeting Hy's Law. And Bob Temple made the observation that if you do have some Hy's Law cases up here, most of the time, not always, but most of the time, you're going to have an imbalance down here with more cases in the active arm, down in Temple's quadrant or Temple's corollary quadrant. So this was the E-dish scatter plot. Now, it was highly successful and continues to be so. We use it all the time. Knock on wood, since 2000, when this thing started to be used, and then other technologies and drug development started to evolve. Knock on wood, we have not removed a drug from the market for hepatocellular DILI since then. Now, but there are problems with E-dish. Some are inherent, some are just how it's used. So one is how it's used. So like any screening tool, or any tool really, it's only as good as the data completeness. And E-dish is a screening tool. I like to say it's like a fishing net. You want it fairly tightly woven, and you want it pretty big. So this was an application we were looking at, and we started to notice there were cases identified in the adverse events tables and narratives that we said, well, where are they? They aren't plotted up here. And so when we went back and we asked, it turned out that the outside labs were not included in the database, and such that this is what was generated. When the outside labs were included, this is what happened to Hy's law, the Hy's law quadrant. And this is a pretty stark example, so that's why I show it. So, I guess if there's a couple slides you want to remember, this would be one. When you're building your outputs, we want those outside labs. You want them in there too. And ironically, see that's exactly the labs you want, because if the worse the injury is, the more likely the patient or subject's going to end up in the local emergency room and even get admitted there and cared for there through the drug injury. And if you don't get those outside labs, none of it will show up on the plot. So again, I would emphasize, and like I said, this is a stark example, but we continue to see it where, the initial one, there's one or two in Hy's law. And then when we go back and say, hey, did you get the outside labs? Now there's 4 in Hy's law. And where there were 10 in Temple's, now there's 20. So I can't overemphasize this enough. And John Sr. was the same. He said, they really need to put all their labs in. If you're going to use this as a screening tool, then use it correctly. I like to say, it's, a fishing net is so good. You wove it really tight, but then you came along, cut a bunch of holes in it, and then threw it out in the water. It's not doing what it was supposed to do. So that's one thing about sort of a caveat about E-dish, and that's more of a user and programmer issue. The other thing that we've been looking at, and we're not sure how this is going to play out, but I brought this slide in because I do want you to think about something, which I'll get to the other slide, is the upper limit of normal, as you may or may not be aware, has grown to be widely variable over the years. And this is data, this is our data, 5,901 upper limit of normal data points. So these are not actual ALT values. These are just upper limit of normal data points that was given to across, I think, like 180 studies over a year span or so. And a violin plot showing the variation. So the lowest upper limit of normal that was reported to us was 15. The highest was 80. But even in this middle range, in the violin, where most of the upper limits are, there's still a wide variation from 55 to about 30. And this coincides with, my experience. When I started in internal medicine as a resident, I remember the ALT upper limit of normal was like 25 to 30. And then over the years, to the point where when I left UNC, their upper limit of normal is 72. And so I'm sure, and everyone is pretty certain that this is, the background fatty liver disease and how labs are choosing their, quote, normal population. And so this is on the radar of several societies and research groups. And this is just one. The ACG came in with this idea that, or this guideline that said that, true upper limit of normal for males is 33 and for females is 25. So, down here, not up here. So I told you that EDISH is based on times upper limit of normal. So this potentially has a lot of impact on any EDISH plot that you use. And we did look at this for this same data. And we looked at how this is both placebo and active arm, and how the numbers change. So the dark blue bar is if you use trial reported upper limits of normal. And as you can see, as expected, if we move it to 33, fix it, and then redo the plot, there's a large jump in the number that enter temples. Also a jump in Hy's law, although not as dramatic. And so we're not clear on what this really means. Is this going to be a problem or not? We have some data that says it may not be as big a problem, mainly I think because it affects both the placebo and the active arms. So the question will be is, so what? If we do fix it, do we see an imbalance, or do we pick up true blue Hy's law cases? And we're still working through that data. But the only thing I would say, that right now for all of you developing trials, I would ask you to ask yourself one question, is that the lab you are using, does their upper limit of normal fit your target population? Most of the time, I think it'll be okay. But there are certain instances where I've seen where it's not okay. And I give the example of you're developing a drug for women who are in their 20s and 30s. You can imagine some diseases where that might happen. So for example, and by the way, this disease makes them sick, so they don't gain weight. So they're going to be lean females, 20s to 30s. So for example, my daughter, who's in her 20s, her ALT runs around 8 to 10. Now, if that trial is so written and she enters a trial, it says, we're not going to do anything with the drug until you hit 5 times upper limit of normal. And the lab you're using, use an upper limit of 50 or 60. That means my daughter is going to stay on that drug. Nothing's going to happen with it, maybe some increase in surveillance. She'll go from 8 to 300 because that's 5 times upper limit of normal. But for her, 8 to 300, you do the math, that's what, 10, 20 or 30 times her baseline. So again, I think it's just a matter of just being cognizant of this issue. And if your population is such that it normally runs, like females run lower, lean people run lower, then it may be worth looking at. And you can run just like we did, run the data both ways, fix it at one of these published normals that would be maybe more befitting of your target population. So that's sort of the message of this slide. Okay, so on to the sort of the large part of the talk is, so clinical trials with abnormal baseline liver tests, they are expanding for 2 reasons. One is there's just increasing interest in developing drugs for both acute and chronic liver disease, autoimmune hepatitis, alcoholic hepatitis, biliary atresia. All of these now have, and I could go on, there are many drugs in development now for these disorders. The other issue that we're a little concerned about is that if you're treating things like diabetes, obesity, and you really want to capture your marketed population, well, you might loosen up, and rightfully so, the ALT criteria. Maybe you'll let in patients with 4 times, 5 times upper limit of normal, as long as they're otherwise stable, and they have stable fatty liver disease. Well, on an EDISH, those patients are already going to be in temples. And for these patients up here, alcoholic hepatitis, autoimmune hepatitis, they might already be in Hy's law, already up there in the quadrant at baseline. So how does EDISH work when the patients start in those quadrants of interest? Well, the answer is it doesn't work. And therein lies the problem. So when I came to the FDA about 5 years ago now, I heard about these trials coming in. It was really exciting. But I said, I was given the DILI task, and I said, wow, how are we going to deal with this? So people were working on it, but the only thing that they thought, and it's a logical thought, is why don't we create what's called an MDish and base it on baseline. So there are problems with that. And to point out those problems, I'm going to step back to the EDIsh, standard EDIsh. And I do this for a couple of reasons. One is, if we're going to solve this problem and deal with abnormal baselines and create some new tool, I would argue that it has to do what EDIsh did for us. And there are 3 functions of EDIsh that you need to keep in mind. And I also present it to you because I would like to tell you how I look at EDIsh and encourage you to look at EDIsh this way. Even in trials where you have normal baselines, standard trial, we're going to use EDIsh, you're going to use EDIsh. But look, do this. And that is, I always look at the first thing I do is I look at the shift. So I do not jump to the 4 quadrant categories. And I mock this up purposely without those 4 quadrants. Because a scatter plot has the greatest power when you don't put those categories on it, right? You just want to look at the overall shift because it has the most power, most data on it. It lets your eye see everything. And here, as you can see, the drug is a little more to the right and up, so more east and northeast, if you will. So right off the bat, I'm thinking, yeah, maybe there's something going on here because I'm also leveraging all this other data down here because I haven't let my eye jump right to those 4 quadrants yet. So I always say, look at the shift first, try to block out those 4 quadrants. After you've done that, then you can look at the 4 quadrants. And this is what people usually do with EDIsh. They jump right to step 2. And I give you these 4 quadrants. And I've already gone over what they meant. And then the third functionality, which is arguably one of the most important, is it creates a triage for us and for you. So we're going to have to go look at these cases, and you just don't want to starting over here, going one by one. This way, you can pick off the cases, these would be highest priority, moving down and to the left. So these, all the ones circled, would be, so to speak, high priority cases for us, and it helps us with our workflow. So this is what you get from EDIsh: normal, standard normal, near normal, clinical trial baselines are normal. But as I said, when you move into the realm of abnormal baselines, then there's a problem. Like this patient here, well, they're in Hy's law, problem. Well, you don't know. That patient could have been there the whole time. They may have started right there and nothing changed. So this patient's fine. They could have even improved. They could have started over here and moved this way. Or they could have worsened. They could have started down here and moved up here. You have no idea because EDIsh doesn't capture that. Same thing with temples. So this was the problem when you have an abnormal baseline for these liver blood tests. And as I said, one of the things to do was, they said, well, logic would say, well, why don't we just peg every patient on their baseline value? And this isn't what we call an MDish. And again, times baseline here, times baseline here, 3 different arms. I will argue to you that the only thing you get from this is the shift function. You lose categorization and you lose subject identification. For example, subject A and subject B, 2 different arms, they plot to the exact same location. Why? Because they both had their ALT go up 5-fold, and they both had their bilirubin go up 4-fold. So they're in the same location. Yet clearly subject B is sicker. This subject should be in a different category and should be prioritized to be looked at. And yet they plot to the same point on the graph. So you lose categorization, you lose subject identification. I'll give you one more example. Arguably the sickest patient started with these numbers, so typical of an alcoholic hepatitis trial. And this is their movement, went up nearly 2-fold, or 3-fold, excuse me, and then nearly 3-fold here. Yet they plot down in this, what I used to call the quadrant of no concern down here. And yet here they are, and they are sicker probably than either subject A or B. So MDish is okay. You can look for shift from baseline, but do not try to categorize and do not try to subject identify. In fact, I would encourage you, if you are generating MDish plots, do not put these 4 quadrants in. And certainly do not label them as Hy's law, temples. That would be incorrect. Those quadrants and those labels do not apply and are not useful in an MDish plot. So this is, where we were left with, the past 2-3 years, and we've been thinking about this for a while. And what we decided, the problem is the missing variable is where the patient started, right? In a standard e-dish, everyone starts down here, and you just let them drift away. But you have no idea, as I pointed out, where these patients start. Now, someday, maybe you guys are, I'm hoping you guys are coming up with some interactive graphics. Maybe it'll be like a video, and you'll watch patients move across these quadrants, or this grid. I don't know. But for now, the idea of migration stuck in our head, and that's what led us to think about a Sankey plot. So this is study 1. It's an actual trial where the patients had grossly abnormal liver baseline tests. Placebo on the left, drug on the right, baselines in the middle. So we categorized every patient based on their baseline values. And then we watched them migrate during the trial. Placebos went from right to left. Drug went from left to right. As you can see, this trial clearly had a lot of abnormal baseline tests because a quarter in each arm were already in Hy's law. We've orientated this so the upward and worrisome migrations are both upward in direction, and we colored them red. So again, going back to those 3 functionalities, what I would argue is, we meet the first one, which is visualization. I mean, you can look at this, and I would say, I think the placebo is doing worse here because we have bad migrators more on the placebo than on drug. And then as far as categorization, yeah, these 2 categories would be the ones I would be most interested in because they're new arrivals in Hy's law. They came from somewhere else, and there they are. This isn't to say we don't want to look at these Hy's law cases that stayed there. They could have gotten much worse, and I'll come back to them. But clearly these are different. They weren't jaundiced before, or this, I'm sorry, Temples was not. This was cholestasis, and they moved over. And then maybe the next priority we look at is the ones who moved into Temples. And you can put numbers on these using a cross table. I'm not sure. Hold on one second. I'm not sure why my bar is showing up. I'll have to leave it. Anyway, so this is a cross table. Patients on drug over here, placebo over here. Where the patients, the category they started is on the row headings. Where they end up is on the column headings. So if you think about it, on the diagonal gray, those patients stayed in place. So 6 were normal, and they stayed near normal. So that's this band here. 15 were in Hy's law to begin with, and they stayed there. So that's this band right here. But there were 2 and 4 or 6 that were new arrivals, and these are these in this category. And you can compare them side by side to the same categories of new arrivals on the placebo side. So you get somewhat numeric and also a visualization of shift. I'll give you another example. We looked at another study as well. This is quite a different study, actually. The enrollment, it's cut off here, I'm sorry, 254 and 765, so many more patients, and it was a 3-to-1 randomization. And here, when you look at this, again, visualization of, is there something going on? That's basically what you're asking. Here, there is something going on, and it's worse on the drug side. The red migrators, there's more of them, and a lot more of them, actually, than on placebo. So, again, this one didn't have quite as many as a abnormal baseline lab tests, but there were enough. There was about 10 or 20%, no, about 10% we were in Temples to begin with. And you are able to get categories of, for example, those that arrived in Hy's Law, newly arrivals. And we could look at Temples as well. And again, get this cross table of the counts. Here, there were actually no patients came into Hy's Law at all, whereas here there were 5. Now, so visualization, categorization, there's that third function. How do we go after these cases? Now here there's only 5, so we just probably gather them up. But there could be, and there will be, scenarios where there might be 20, 30 new arrivals into Hy's Law. So how do you decide, which ones to go after, or which ones are more important to you? So for this third function that we want to replicate from EDISH, we reached back to a paper that was published by our group a little bit earlier last year. And this is case identification by composite plotting. It's a little difficult to digest a bit, but I'll walk you through it. First of all, these are 4 discrete MDISH plots. MDISH plots based on baseline for each patient. And what they did, or what we did, we grouped them by every patient that ended up in cholestasis here, every patient subject that ended up in Hy's Law or up here, ended up in Temples, and ended up normal, near normal. And then we color-coded them by where they started from. So if you remember, let me enlarge. Let's just say we wanted to look at the ones that ended up in Hy's Law. So if you remember, there were 4 and 1. There's the 4, and they're the green X's. They came from normal, near normal, and ended up in Hy's Law. And there was 1 who came from Temples, and that's the blue triangle there. So there they are. So if there were like 20 of these, then I would move from right to left, because these guys are up here or downward this way. These guys up here will be moving their baseline more than anybody down here. And in fact, remember I said that had there been patients that started in Hy's Law and ended in Hy's Law, I hypothetically gave you these. I would barely look at these, right? Because actually they improved. They're less than their baseline values. These are the baseline cutoffs. This guy I would definitely look at. He moved his baseline quite a bit. So this is a way to even look at and triage the ones who stayed put in a bad quadrant, Hy's Law or Temples. So again, this was a way to look at visualization. Is there a problem or not? Categorization, okay, which groups of patients are we going to focus on? And then the third function, how are we going to look within this group? How are we going to look at them, and in what order and what priority? So we're hoping that this kind of a process, although not as elegant, as simple as EDISH, will help with our workflow and our triage and organization of how we look at these events in these kind of trials. The other plot that I'll give you is, this is called a modified waterfall plot. Now, I will point out that this is study 2. So again, the same study I just showed you. And the reason we like this plot, or we thought of it, is if you remember in study 2, there was hardly anybody that was jaundiced. Nobody in cholestasis, and really nobody in Hy's Law. So this trial actually you couldn't enroll if you had hyperbilirubinemia. So what that means is that we really have only one variable we have to be concerned about that's abnormal coming in the door, and that's ALT. The bilirubin, we don't have to be worried about it. And then it puts it back on a 2-dimensional plot just with one variable. So what we did was we plotted every patient, their baseline value along the black line, from lowest to highest for the placebo. So the patient who entered this trial on placebo, the lowest baseline ALT was right here, down here, and the one with the highest is up here along this black line. For the drug, we did the same, but we just went in the opposite direction, highest here and lowest here. And then we just plotted by a bar their maximum ALT after day 1. And so you get visualization. You say, clearly there's a problem here. You know, there's more excursion up on drug than placebo. Secondly, interestingly enough, this drug worked. It brought these ALTs down greatly. It had great efficacy. So that's that curtain of downward brown, but it also had a little bit of a DILI signal. And then what we did is anybody who got jaundiced, because remember I said they couldn't have significant hyperbilirubinemia. So any jaundice was new. And so we tagged them as green. So now you have visualization and you can do categorization. Maybe you could say, well, I want everybody over 300, and I certainly want anybody with a green bar, or maybe over 600. So you can make your cut, categorize, and go after these cases. You know, ideally we would just have a list and we would just go after these, or you could point to them and it'd tell you the case number. So this is a modified waterfall plot. This one would be an all-in-one. It would take care of all 3 functions, all in one graphic, if the bilirubin has been restricted at enrollment. So you only have the baseline ALT to worry about. So we're still working with these plots. We've worked on it with one more trial at this point, and it seemed to be working okay, both the Sankeys and the waterfalls. So what I did is I walked you through the eDish, its origin strengths. I highlighted some of its failings and pitfalls, like incomplete data. Remember, that's the one where I showed you 2 eDishes. You know, hardly anybody in Iceland, there was a lot after they put the outside labs in. Upper limit of normal variation, I would just ask you to ask that question. Make sure you're not treating your target population that has significantly lower upper limits of normal, that's normal for them. And then I covered the trials with abnormal baseline liver tests and what we're trying to do with those. So I will finish up with some deficiencies in trial protocols regarding DILI risk and cover these 3 topics here. Herbals, just overall case level data reporting and how it would help to get it organized and data collected. And then the value of stored serum, or stored blood, I should say. It doesn't have to be serum. So herbals and dietary supplements. This is really important, and I think that really all trials should really think about this a little bit. So these are 4 fairly large registries of drug-induced liver injury. And right now, the DILIN in the US, we reported out at 20%, but that actually number has gone up even more. We're up around 30%. So of all the cases that come in now, roughly 25 to 30% are due to an herbal or drug and not a pharmaceutical. The Spanish group came in at 20%. China came in at a whopping 27%. Now, I point this out because herbals are so steeped in tradition in China, and they're used so much. And I know that I see a lot of clinical trials these days where sites are being recruited from China or Asia, and this becomes an issue. And then the Latin comes in a little lower at 11%. Clinically, this is an issue because, see, subjects and patients, they are less inclined to divulge their herbals if you don't pointedly ask them about it. And if you don't, and there's a liver injury, your drug will be on the hook, when in fact it was an herbal that the patient was taking that you didn't know about. And even if you do find out about it, do you want that noise in there and the sort of angst and havoc that it creates? Because then you're going to have to report it, work it up, do all those things. So what I usually recommend is I say, I would, if you cleanest thing would be to just exclude HDS use, but I know that's difficult and it could hurt your enrollment. But I would say this, limit the HDS use then to stable dosing to 4 to 8 weeks. I like 8, but I knew there was, we had a cancer trial and patients were desperate. They don't want to wait 8 weeks, So, 4 maybe, but make sure it's, they're doing well and they're on stable dosing. exclude HDS products that have well-described hepatotoxicity, like green tea extract, ashwagandha. There's published publications on all kinds of ingredients that are sort of tagged as toxic to the liver. Muscle building agents, anabolic steroids is a big deal. They have a significant signature for bad cholestatic injury. And then no new HDS products or dose increases without investigator approval. I think that's sort of a given. And I would point out that I don't think people know what these do to drug metabolism. We all know about grapefruit, but I don't know that anybody knows what these are doing to your SIPs and things like that. So I think controlling your HDS should be sort of a routine thing nowadays because they are being used so much. Case level analysis. So this comes down to just sort of meticulous attention to detail. This is internal medicine at its best. We rely on the narrative. We want graphic and tabular profiles. I give you a picture of a character in a TV series when I was growing up. Detective by the name of Columbo, who was played by Peter Falk. So he was known for being this persistent attention to detail kind of guy. He said he'd always come back and say, I just want one more question, please. Just answer me this. I just need this little piece of information. So it's one more information request is what this is all about. And so the way that the data comes to us is sort of non-standardized, and some you'll see if you're involved with a liver injury with us on the DILI side, on the DILI team, you'll see that we ask for certain things in a certain way. And firstly, what is a good narrative? Narratives vary, in my experience here, quite a bit from really quite incoherent to really excellent. I color code these for a reason you'll see in the next slide, but we want good baseline data. We want well-documented time course, signs and symptoms, and address the alternate causes. The big four are these first four, and then these others come in behind them. Let me say comments on this time course issue. What I like to think of a narrative is it's like a bedtime story to your child. And it's like, once upon a time, there was this man, he was 65, and this happened, and then this happened, then this happened, then this happened. It's not like a movie or a novel we read these days. Like, there's tons of flashbacks. Like, oh, by the way, 3 months ago, he had dark urine, or, he was taking this medication since 2 months ago. It gets incoherent and hard for us to analyze, and I think would be the same for your reviewers. So very methodical. And I think ChatGPT will help here. I think if you can program it to say, listen, everything chronologically in order, please, so you don't jump around. So this is a mock-up of what a narrative would look like. It's not from GPT, although we have that now at the FDA, we have access to something similar. But this is sort of a mocked up narrative to just make a point where, the yellow part is up front. We want that baseline data. We don't want baseline data being mentioned to us way down at the end of the narrative. And then you're going to tell us that the patient had alcoholic liver disease in the background. No, don't do that. Just give it to us up front. And then it's just chronologic, day 1, day 6, day 18, day 140, day 146, and so on. And then this happened. And then the workup, I think it's good to just put it all in one place. I think that's reasonable. If you want to stick it in here when and where it happened, that's fine. But again, it's hard to pick it out when it's all jumbled in there. A line graph. So you'll see us ask for this. I show you this one because this line graph actually ended up, so to speak, exonerating the study drug. So what we had with this particular case is we had tabular data of the liver tests, and they told us when the drug started and stopped. And when you looked at the tabular data, it looked bad for the study drug because clearly, the enzymes went up, got jaundice, clearly a Hy's Law case. And then on another table, they had the concomitant medications. So what we said, well, wait a minute, there's this statin, let's just see, and we graphed it out. And what you see is that you didn't pick this up on the tabular data, is that the enzymes actually came down, and that's what everyone focused on, but they actually started going up again. And the drug's half-life is not that long. And it was only after the simvastatin was stopped that things really got better. And so this was enough for us to say, hey, the study drug could have done this, but you've got a pretty strong competing etiology here. And it took this drug off the Hy's Law list, actually. So my encouragement to you is if you're going to look at these cases, these line graphics, I think this is true of clinical meth, out in the hospitals when we were using Epic, where I was, we could get these kind of graphs. And it was very, very helpful for trying to determine what was going on. So a line graph. Excuse me, sorry. So this is an example of our information request is sort of standards, 3 pages. So we give you, what we'd like to see in a narrative, kind of tells you what we'd like. We actually put in a table of the evaluation testing that was done. Let me be clear. This is not to say that you need to do every single test, but we want to know, yes or no, did you do it? Or did someone do it? And what was the result? When was it done? We would also like a table of absolute values, not times upper limit of normal. As I mentioned before, if you just put an ALT that was 5 times upper limit of normal, I don't know if that is 5 times 3, 150 or 5 times 60 or 70 over 300. And to a clinician, that's a big difference. An ALT of 150 versus an ALT of 350. So we do want the absolute values, not times upper limit of normal. And we ask for it in this format. As you might imagine, we want CPK LDH to see if it's hemolysis or muscle injury, that kind of thing. And then here's that line graph we asked for. So this is, if we're really on the hunt, something's happened and there's been a significant liver injury, and we're looking at these cases, we're gonna want or ask for and require these kind of data. I grant you that sometimes you can't control it. Maybe the investigator or the local site didn't collect all this data, but we want to know, yes or no, did you get it? Did you not? So we can go on from there. Now I'll highlight hepatitis C RNA for a reason. It sets me up for the next 2 slides, which are actually my final slides. Hep C RNA is often left off in the evaluation, and it should not be. It actually, when you're looking for acute hep C, it should actually surpass the hepatitis C antibody. And here's why. There is no antibody IgM equivalent for acute, like it is for hep B or hep A. There's no such thing for acute hep C. They don't have one. HCV RNA can be positive before the anti-HCV seroconversion. So that's why hepatitis C now, RNA PCR testing is recommended for acute hepatitis C by SIOMs and multiple liver and GI societies. So in other words, your standard screen for viral A, B, and C, it should be serologies, antibodies for A, serologies and antigens for B, and hep C RNA for C. I would rather have that, frankly, than getting necessarily the antibody. You can get both, but just get the RNA for sure. And let me show you this next slide. This sets me up for the next slide, which is my final slide about the importance or value of storing a blood sample when a liver injury occurs, and storing as many as you can get, actually. So this is a case of an adult male. He was from Ukraine, and he was getting a study drug, 1st and 2nd dose. And he had these elevations in ALT, AST, and he became jaundiced, bilirubin up to 5. So met Hy's Law. And, it was a little unclear to us because we knew a little bit about this drug that was in trials, and it's unusual for it to cause hepatocellular injury. So we asked, and they were fortunate. They did, believe it, even in Ukraine, when this was, I think, just before the war started. they had stored serum. And lo and behold, see the black here are negative. This is the antibody. He was negative at screening, and he was still negative at the time of the entry. But when they went back and tested for RNA, he was positive coming in the door. He later seroconverted, developed the antibody, and was persistently hep C RNA positive. So this is classic for acute hep C. And he got infected shortly before screening and coming into the trial, and then manifested. And this is, in retrospect, this is pretty typical of hep C, where body's trying to clear it, doesn't quite make it. Hopefully he got treated and cured. So hep C RNA, real important. And in general, stored serum samples, they're great. You can get some other tests, do PK on it, and all kinds of things during an injury. It can be really very critical. So just to sum up, EDIS remains the backbone screening tool for DILI, but EDIS is only as good as the data which are entered to generate the plot. Other plots or methods like Sankey, cross tables, composites, and waterfalls may be needed when the baseline liver tests are normal, particularly if they're markedly abnormal. Then EDIS will definitely fail in its functions. And then key issues, control the herbals. I think that should be sort of the standard, something you might want to look at. It'll pay off dividends down the road. Comprehensive case data collection, I went through that. And then stored blood samples, hep C RNA testing, that kind of thing. And with that, I am done. I'm going to give up the screen here. And let me see, hold on, end slideshow. Yep. Jim, are you on? Did you, yeah, there you go.
Yeah, absolutely. No, thank you very much. It was just an outstanding presentation and brings a lot of important points to the forefront that we definitely need to consider when we're looking at our safety data. Before I turn this over to Barbara and to Cynthia, I just wanted to make one particular comment as it relates to the Hepatic Explorer, which folks may know is one of the ISG tools. It's an interactive eDish graph. One of the capabilities of that tool is to generate MDish plots. And I do want to point out, as Dr. Hayashi had mentioned, that there are some substantial problems in running MDish plots and then trying to interpret the data out of that. So I would caution people that even though that tool has the capacity to produce MDish plots, we would really suggest strongly against using it terribly, given the fact that, as he said, you lose the categorization capability. And you very, very well may likely miss some important cases as well too. So just a caveat about using the Hepatic Explorer's MDish capabilities. That said, we certainly would like to expand that interactive tool, and we look forward to the opportunity perhaps to include composite plots and tables and so forth as a way to sort of move that science forward. But with that said, let me now turn the podium over to Barbara and Cynthia, and we'll get into some Q&A.
Jim, do you want to address the questions in the chat for Paul first?
We could certainly do that. We have a number in here. Let me just scroll back up.
I think it starts with Michael Wright-Colaby. I think the other comments are about we had some noise and chatter at the beginning. Am I seeing that right?
Yeah, I think you're right
Yes,
here.
that's correct.
Actually, Paul, yeah, if you've got access
I
to
can,
it.
sure. So he's mentioning the Pfizer's halting development of an oral GLP-1 small molecule. Yeah, it's a difficult question, a go, no go. Yeah, based on a single case. So to answer, I can't speak for Pfizer. I don't know what their decision process was, but looking from afar, first of all, what I understand is they were in early phase, so they didn't have a huge denominator. Remember I said, if you have like 5,000 exposed and you came up with one high saw case, that might be manageable, right? Not every high saw case dies. In fact, only 10% of them will go that way. But I imagine their exposed population was relative. They were early in phase development. And then you have to flip over to the non-safety side. Like, who are you treating? The patients for, I believe it was for obesity, is huge. So and then there are drugs now approved for obesity, as you all know. So then, is it truly, truly now a completely high unmet need? And then my sense of this, because I did think about this, I saw this too. I don't know. I don't know what Pfizer went through their head, but it's a lot of patients that might go on this drug, and there might be some cases, and then there's another drug out there already. Granted, it's a subcutaneous injection. So that would be my answer. I hope that helped. So yeah, it's not like one case and you're done. It's a much more nuanced decision. What is the optimal window for AST at total? Yeah, I get this question all the time. So in our EDISH plots that we make and generate at the agency, let me just show you this one. So there have been plots made by both the agency and elsewhere where they have said, well, if it's not within 30 days, we're not going to plot it, because it's probably not related. And I said, stop that. I like the 30-day window, but you see, what you will potentially lose is, when you whack a liver really badly, the enzymes go way up, the bilirubin starts to climb. The enzymes will fall, but that bilirubin will continue to have a pretty unrelenting climb because you've crippled that liver. And they may go on to transplant like 8 months later. And that bilirubin peak will actually happen 2 or 3 months after the injury. So the computer will simply look for the highest bilirubin, and it will say, oh, well, it was beyond 30 days, so we're not going to plot it. So 30 days is the general window, and what our programming does is it will circle any case within that 30-day window. But we still want the case on the plot. We don't want it scraped off the plot. I like to say I don't want really any patient not on the plot. I want them on the plot, and we'll figure it out on the back end. So 30 days, 15 is a little tighter, maybe more specific, but we generally go 30 days. It's the window we use.
Let me just interject perhaps
Yeah.
Another question to that. Is it worth thinking about Hy's Law qualifying cases within that time period? So in other words, like you say, the bilirubin may continue to increase over time, and so the peak doesn't really occur until much further out after the hepatocellular injury. But within that 30 days, they've already crossed that twofold elevation.
Right. And you're absolutely right. And that would still get you to capture the case. So I think, like I just said, I think 30 days is a good window. Usually that bilirubin is going to get, if it's going to be that bad, it's going to hit 2 times in that 30-day window. I guess, so my point was, I've seen plots both at the agency and outside where they just said, well, it was outside the 30-day window, we're not going to plot the case. And they wouldn't be on the plot. And I said, don't do that. So, is there any slides supposed to be doing? Okay, I think this is a technical questions. This is a veterinary clinical pathologist asking a question. Do hospitals measure ALT agree on the use of assays without the addition of P? We have found that values on our animals can differ due to the addition. I can't read the P. Is that, I'm guessing that's the vitamin. Pyridoxine, is that what you're saying? This is from Paula Katavalos? Without the addition of P something. I can't read it for some reason. There's something blocking it.
It looks like E5P.
I'm guessing that's pyridoxine. I'm guessing that's the vitamin. I hope that's right. I'm going to answer it if it is. So I think what she's getting at is in the assay, the ALT will be abnormally low unless you add back this vitamin. I think it's vitamin B6. I can't remember. And I think that's what she's getting at. And what I know about that is the labs across the country, they vary. Some do it and some don't. And there's been literature about that. You prefer it if they do it. It's more accurate that they add in the vitamin. And where this comes into play is if the patients are very malnourished, they will have an erroneously low ALT value. And where we see this is actually in alcoholics to come in. And some people have said that's why the ALT and AST ratio is this way. When they first hit the door, yes, thank you. That's great. Yeah, I thought it was. So ALT and AST, the ratio is higher in alcoholics. It's the AST higher than the ALT. And what's been written is a lot of it is sometimes because the alcoholics are so malnourished, the ALT is inordinately low. And through the hospitalization, you'll actually see that ratio normalize because we start giving them IV, what we used to call banana bags with IV vitamins in it. So I think but what I know about the labs is that some do, some don't, and you'd prefer it if they all did. This one is the Sankey plot on plot 1. Yeah, plot 1. This would be this one. How would you use that to contextualize the shifts in the active arm? Oh, I see. So I think what you're saying is, some of these events in the active arm, what happened? Why did they do this? Because it's not as bad as in the placebo arm. And he's asking, is this disease progression? Yeah, it very well might be. I can't tell you what this disease is and what they were looking at, but we do think that that's, in other words, these migrations might be subsets of these that were just natural disease course. And that's probably why the placebos had all these. We think the drug had efficacy, so it actually prevented some of these migrations. So that's, you're absolutely right. So do we have to think about that? And honestly, I don't remember how we adjudicated all these cases. I don't think I should get into too much detail, but some of them we might have said, yeah, this is just disease progression. I hope that answers that question. But we consider them sentinel events. Sure. Well, events in terms of we're going to go look at them, and then we'll find out if we blame the drug or not. What has been the typical time to onset of DILI with herbals? Oh, would it stable? Yeah. No, yeah, you're right. It's all over the place. I only put that 4 weeks down there because I told him to keep it stable for 3 months. And this was an oncology trial, and the oncologist said, hey, these patients are dying. And we can't have them wait another 2 months and on their stable herbal. And people take herbals for cancer and all these things. So that's why I put that 4 weeks. But you're absolutely right. Herbals are all over the place, but generally it's like pharmaceuticals. It's 1 month, 3 months, 4 months, 6 months. We'll see the latency. Barbara's question was, since we don't really know what herbal supplements do to the body, is there a possibility of DDI between herbal and study? Yeah. So Barbara's asking, or Minjun is asking, is there interactions that we have to worry about between herbals and pharmaceuticals? I can't think of an example. Grapefruit, everyone knows that one. But and then St. John's wort is another one. So I'm sure the potential to affect DDI in your pharmacokinetics is definitely in there. And I worry about that. the herbal industry is a billion-dollar business. And, most herbals are probably fine. I dare say some people on the call may be taking some dietary, herbal thing like turmeric or something. But in a clinical trial where you're looking for clean data, I just raised the question, you may not want that noise. So, what DILI time to onset on a chronically administered study drug would make a causal relation very unlikely, greater than 6 greater than 12? Yeah. I think, greater than 12 months, yeah, we're getting less and less inclined to blame the drug. Although, as we all know, there are some drugs that take, believe it or not, years, nitrofurantoin and minocycline, those kinds of drugs. But you're absolutely right. Greater than 6, we're starting to get a little, well, maybe not the drug, but we would still implicate it at 8, 9 months. Greater than 12, I'm going to be looking at it, a little more closely to say, am I really going to blame this drug? And I think that's reflects what's done clinically out in the post-market world, and we sort of mirror that here. Okay, in your publication, this is the one that looked at composite plotting for patients with underlying liver disease and who are in the temple and high quantity baseline. Would you consider them to be that to be a clinically important shift from baseline? Oh, is the FDA receptive to sponsors using your new approach in lieu of the SMQ approach? Yeah. I can't. So the first question, consider to be a clinically important shift from baseline. Who are on template baseline? I think I sort of said this. If they are in temples in Hyzlod baseline, and they shift within that quadrant, yes, it sort of depends on how much they shift. Like if they came in with a bilirubin of, 2.8, and now it's 17. Well, they're still going to be in Hy's law, but that's bad, and I'm going to look at that. And ALT similar. If they came with an ALT of 180 and now it's 700, we're going to look at. So that's where that composite. We're going to look at multiples of baseline. If you're going to try and pin me down and say, well, if the baseline similar to, a normal baseline, the baseline more than triples or more than doubles with a more than doubling of their bilirubin, I'm going to get upset and I'm going to look at that case a little more concerningly. And is the FDA receptive to sponsors using your new approach? I can't really comment on that. We're early into this, and it's not a policy that we're doing. Having said that, I suspect that if you're having a big problem with abnormal baseline tests, you're probably going to be coming in with an application to the Division of Hepatology. And in our division, we would look at that data receptively because we obviously generated these approaches, but they are not an official approach in any way. That's all I can tell you. They'll be mentioned in the guideline, but they won't be pushed. For autoimmune hepatitis, will you consider to use 5 times, I'm not sure, 5 times half-life time frame for monitoring? If not, is there any way to monitor the onset? So I think you're getting at is when can you stop monitoring after 5 half-lives? Yeah, that's an interesting question. I think the only problem I have with autoimmune hepatitis, I worry a little bit that they're going to have like this sort of autoimmune-like hepatitis from a drug. And there, I think that the delay in the onset could be, because it's an immune reaction, it could be delayed. But I think in general, we're going to, consider 5 times half-life. We're going to say that drug's gone. And then we will, again, look with a more jaded eye that we would be blaming your drug. Now, whether to monitor, in autoimmune hepatitis, I would suggest I think you're going to monitor anyway, longer than after the drug, because they fluctuate so much. You're going to want to know if they rebound off your drug at 2 months or 3 months. So I think in autoimmune hepatitis trials, I think you're going to need longer follow-up because it is a chronic waxing and waning disease. And what you don't want to see is a rebound, right? It's like prednisone. We see that all the time. We lower the prednisone and then they do okay, and then they have a relapse pretty quickly or not so quickly. I hope that answered that question. While comparing data between treatment and control, how much do cases and temples correlate, contribute to the DILI risk of drug? As the DILI risk may be reversible. Yeah. If the cases show reversible injury and the ALTs are not exorbitantly high and you have nothing up in Hy's law, then yeah, we're gonna say, well, it has the potential. We may, get into, and we attribute it to the drug. We'll get into labeling, but would it be like a showstopper if the exposure population is fairly high? No, we would just say, yeah, we see that all the time, and that's what we call adaptation. How much do cases and temple score contribute to this? It contributes, but as long as there's no Hy's law cases and they are getting better, it's not going to be a huge showstopper. Labeling, it'll come up in labeling probably. I hope that answered that question. And that's it. That's the questions I'm kind of at the bottom of the scroll bar here. Anything else?
That's great, Paul. I'll just make one comment and then let Cindy speak. But our aggregate safety assessment planning task force, has a template where we have what we term safety topics of interest. And as part of that table, we have a category of events called other safety topics of interest, which we define as those events which you don't have any reason to believe that your product has any causal association, perhaps, but they're of traditional regulatory concern or of concern based on the epidemiology of the patient population, for example. So DILI is a very common example I give of an event that should be under the other safety topics of interest for nearly every product, regardless, as I said, of whether you believe there could be a causal association with liver injury from the onset of your development program. Because we know that when you submit a product for consideration of approval, that the regulatory agencies, especially FDA, they're going to look at your data very, very carefully. And you really need to, as Paul has mentioned, have all of the pertinent information collected on these cases. I mean, to go back 2 years later when a case was, reported before your filing, and to try to get that information a couple of years later is just not a good idea, generally. So you really need to have in place how you're collecting supplemental information. Paul showed you some of the supplemental information that they would like to see. And so I'm a big advocate on having supplemental case report forms that you require the sites to complete for cases that meet certain criteria, like any serious events, any events that have a transaminase level over a certain level per your patient population, or a bilirubin over a certain level, or any discontinuations due to liver-related events. You can map that out in your protocol, what you're asking your investigators to do in terms of which cases to complete this supplemental form. But I definitely think practically every trial should have that, and not wait for you to see that your product has a problem potentially and then start implementing all this. I mean, I think that it should just be standard, for development that you're doing this. and also that you're routinely monitoring with some of the plots that Paul showed, looking for any evidence of an issue, in the product that's while they're under development. And certainly at the time of submission, you need to do detailed analyses just to make sure you're not picking up on anything. And I can't emphasize enough that even if you don't see transaminase changes in your trials, that doesn't mean that the regulatory authorities won't get fixated on one case in your whole database. And that can lead to a warning in your label, just one case. So you really need to be on top of it and really be making sure that you're monitoring this in real time, collecting information in real time on cases that might be of potential concern. So that's my main comment, and I'll let Cindy give her comments.
Sure. Thanks, Barbara. I don't have a lot to add other than supporting that. Definitely support the routine look following a ASAP or similar process to in your ongoing safety review to be looking at some standard approaches for evaluating, or I'll say screening, if you will, for events that require further look. And there are, as Jim has mentioned, there is a open source tool available, Safety Graphics, which we've presented on before, or others have presented, that is very helpful in exploring data. That is a really nice supplement, at least from my experience at our company. It can be a supplement to those GXP TFL deliverables that we do. So first off is standard safety looks. So if you standardize your routine look at safety data to include a look at potential hepatotoxic cases, and have a means of looking at those in a very efficient way through data exploration using a tool like a Safety Graphics or something similar. If you have something different internally, it can really help you identify things in real time, create those patient profiles, those narratives, and really navigate through your data and understand it well before you ever get to the point of submission. That allows you to be able to discuss it at length, or if need be, add additional data collection points for studies if needed, if not already planned for. So I would really advocate the use of graphical tools like ISG. I know that maybe depending on how your company, the policies of your company, it may or may not be easily, but I would encourage everyone to partner with your statisticians or programmers to see how you can access a visualization type tool like Safety Graphics if you don't already have access to that.
Great. Thank you very much, Cindy. We have a few minutes left before we conclude, and so this might be a good opportunity, Paul, to perhaps address a couple of other topics. One being I think the value of the R or the NR calculation as opposed to absolute alkaline phosphatase values, and maybe if we have time, also talk about when the baseline sort of gets reset. And I think it was the bathtub plot that was a good example of that. So,
sure. I see Will Stream came on video. Will, did you have a question, or are you okay? I know Will.
Yeah. Hi. Oh, can you hear me?
Yeah, I can hear you.
Okay, great. No, I have a couple of questions, but I don't want to, hijack anybody if you want. I guess just two quick ones. And the first is I know that you and Paul Watkins and some other people have been working on the whole idea of area under the curve for ALT, how fast it goes up, how fast it comes down, as being significant in terms of really adjudicating how much injury there is. And so I'm wondering what the status of that is. And then the other thing is, in the newest review article, that you had a nice kind of cholestasis E-dish plot that you showed, that I think used data from a from a study from obeticholic acid in MASH. So I'm just curious about those two developments. I know they're maybe in progress, but your thoughts about how they might evolve and be used.
Sure. What Will is referring to is that, EDISH is based on the peak ALT value. Well, logic would serve that maybe it's the area under the curve that will more accurately quantitate the liver injury loss. So Paul Watkins came out with this P-ALT equation several years ago. We are looking at it. I can't tell you much more than that. We have a fellow working on it. I can only say it's difficult because the data we get, even in clinical trials, doesn't always create a nice curve. And so it's always a challenge going from a mathematical model to actual real world stuff. But we are still working on it. Right now, we don't have anything to suggest that it's better, or we're going to switch to that. As far as the cholestasis plot, so what Will is referring to is an EDISH plot, but does not really address cholestasis, or cholestatic liver injury, which can be severe and even fatal, where the alk phos drives the liver injury in terms of the liver blood tests. And so we have used a plot with the x-axis maximum alk phos instead of the ALT and ST. If the guideline goes forward, if we can, it will be mentioned and it will be discussed, sort of in the same vein as the article that was written. I was one of the authors. It's just to point out that it can be useful. It'll certainly shows those imbalances and shifts. And I justify the quadrants that we cut in that article. But I'd also point out there is no Hy's law for cholestatic injuries. So you will be out in uncharted waters when it comes to that. But we will use it for categorization, looking for cases. It's just a matter of what does it mean? I mean, I don't know that they have a 10% mortality risk. I don't know that. I hope that sort of addressed your questions. Well, okay.
Yeah, thank you.
Sure. So I'll quickly cover this. I'm not going to go to presentation mode, but this is the issue. Bottom line is we're trying to get away from using, I think we will be, this is just me talking. I'm trying to get away from using the criteria for just elevated serum alk phos of it has to be less than 2 times upper limit of normal. So this is straight from the current guidance that, as I said, it's worrisome to have too high salt. And the way it's defined is no initial findings of cholestasis, elevated serum alk phos. And it goes on to give an example of less than 2 times upper limit of normal. So in other words, if the alk phos were over that, then it wouldn't meet Hy's law, so to speak. And the trouble is that the alk phos does actually go up in bad hepatocellular injury. And that was brought out by something called an R value, which is this. And this is in the post-market literature, where an R value is actually the ratio of the ALT divided by the alk phos corrected for upper limit normal. Greater than 5 is now considered hepatocellular. And just the N part just means you take the ALT or AST, whichever one drives a higher value. So post-market data suggests this is better than having a hard ceiling on that alk phos if it can't be higher than 2 times upper limit of normal. So we looked at the data because it's unclear whether that's true in clinical trials. And it turns out it's true. The R value is better. This is in our experience, the positive predictive value, which is essentially Hy's law, and you want it to be around 10%. In our experience, the NR value was closer to 10%, whereas this criteria was not as high as 4%. And more importantly, we did have some fatalities, about 3 or 4. The NR value picked up all of them, where the alk phos only picked up 1 in 3. And then 1 in 4 if you include the acute liver failure patients. So in other words, it's closer to Hy's law, and it's more sensitive to picking up the fatalities. And these are the fatalities well, 3 fatalities and 1 acute liver failure. This patient survived. But you can clearly say these are hepatocellular. The AST and AST were in the thousands. And unfortunately, see, the alk phos tends to rise a bit. So only one of them stayed below 2 times upper limit normal, whereas the R value was well over 5. So we are sort of suggesting so the R value definitions is superior. Either replace it, or if you can use both, but it's either or. It'll make criteria for Hy's law if it meets either one, and that's sort of where we're landing with this. But yeah, the R value is what the R value does in the post-market setting seems to be the same in the pre-market setting. And that's it, Jim. You only got a couple minutes, so I think we're done, unless you have other ideas.
Great. Well, we have come to time. I want to thank Doctors Hayashi, Hendrickson, and Cindy for their participation in this webinar. We've learned a tremendous amount. I hope you all have some great takeaways for this. I just posted in the chat if you'd like a copy of Dr. Hayashi's slides. Feel free to email me, jbuchanan@covalence.com. Make sure that you get a copy of those. Thank you, everybody, for your participation, and we look forward to the next scientific webinar. Thank you very much.
Thank you all.
Thank you.
Thank you.
Thank you.